Hi everyone, my name is Shannon Reaver and I work for Facts based in Stellenbosch. And today I'll be speaking to you on the topic of combating honey fraud with focus on testing challenges and regulatory shortfalls. Honey fraud is a major global issue, with honey being the third most faked food in the world behind milk and olive oil. Acts of honey fraud can generally be classed as either adulteration, which is the addition, substitution, or dilution of honey with products that are not made by honeybees, or misrepresentation, which is the masking or mislabeling of botanical and or geographical origin of a honey. Adulteration definitely makes up the bulk of honey fraud cases, but what about misrepresentation? This presentation is largely going to focus on the current legislation for honey misrepresentation, the available tests for determining honey origin, and also discuss whether this topic should be a focus area in the larger honey fraud space. When we speak about misrepresentation, we can refer to botanical origin, which is the plant source that the honey originates from. And here we can talk about monofloral honey, and that's honey that's coming mainly from a single plant species, or multifloral honey, and that's honey that's coming from multiple plant species. We also speak about geographical origin, and this is where the honey originated from. Monofloral honey tends to have a higher demand and economic value compared to multifloral honey, and this is on account of its more appreciated and distinct flavor and aroma. Single geographical origin honey also tends to be more valuable compared to blended honey that contains honey from various different geolocations. So there's definitely economic motivation to make fraudulent claims regarding origin. The extent of honey misrepresentation worldwide is unclear, but to get an idea, over 3 times the quantity of Manuka honey that is produced in New Zealand is sold worldwide. This highlights the major problem of misrepresentation. This honey may be pure honey, but it's not made in New Zealand and not made from nectar collected from the native tea tree bush. So you can't call it Manuka honey, it's honey origin fraud. Honey misrepresentation does not have any health implications, but it can have considerable economic implications, as it may affect consumer perception of premium products. So what does legislation currently say about honey fraud? Local regulations and international standards provide compositional requirements for honey, which largely serve to protect against adulteration. But there is a shortfall in current legislation regarding honey misrepresentation. If we look at South Africa's honey regulation, R835, as well as the Codex standard for honey, you'll see that all the listed parameters are mainly focused on checking for adulteration. None of these parameters specifically look for any kind of misrepresentation. Beyond the listed parameters for assessing composition, R835 indicates that a honey can be marked with its floral source or geographical origin, provided that a true reflection of the kind of honey is contained therein. Similarly, the Codex standard for honey indicates that honey may be designated by its geographical region or its floral source if it comes exclusively from that area or that particular source. But nowhere in these documents is there any kind of guidance on testing for origin authenticity. Why is this? So if we look at the available official methods of analysis of the AOAC, you will see that there's no OMA listed for determining botanical or geographical origin of honey. So it's difficult for regulations to actually give guidance on testing methods if there are no testing methods that are officially available. Earlier this year, the AOAC put out a call for standard method performance requirements on a non-targeted test for honey fraud evaluation. This test could, in essence, focus on either adulteration or misrepresentation or a combination of the two. There is currently no standard method that can be used by the global market for effective honey origin control systems. So the focus of this presentation will be on the potential methods that could fulfill this SMPR for misrepresentation testing. Origin testing has been a major focus area in honey authenticity research, with 65% of current research articles focusing on techniques to investigate and prove origin. The reason for this large body of research is that honey origin testing is very complex on account of the great variation that exists within honey. So this variation needs to be completely captured in any kind of model or test to account for all possible honeys. Whether this is possible, I don't know, but let's look at what techniques could be candidates for this type of testing. Currently, several tests are available for investigating honey origin. The traditional approach to identifying honey origin is by microscopic pollen analysis. Pollen analysis is often supplemented with physicochemical analysis and sensory analysis. Although this method is well established and has been performed for many years, it does have various drawbacks. Pollen analysis has a very low sample throughput, so it's not always feasible for commercial application. It also requires experienced personnel and a comprehensive collection of pollen. Also, you need pollen to actually be present in the honey for the method to be successful. This is tricky because some pollens are naturally underrepresented in plants, and therefore their abundance in honey will be low. Honeys also may be filtered, which reduces the total pollen count. Moving on to more modern techniques that have been explored, chromatographic methods are currently the most researched approach for determining honey botanical origin. Phenolic compounds are used as botanical origin markers, as the phenolic acids and flavonoids in honey are directly transferred from the collected nectar. Isotopic methods are generally well established for detecting honey adulteration, but it is also possible to use these methods to distinguish geographical and botanical origin, but you use then different stable isotope ratios compared to honey adulteration. However, isotopic techniques have shown that it's better to combine them with other techniques to improve reliability. Spectroscopic techniques are generally useful for investigating both botanical and geographical origin. They generate spectral fingerprints and capture various compounds in a single run. Specifically, NMR and NIR have been very useful for honey authenticity studies. These methods all require chemometrics or statistical modeling to extract relevant information. So they work on pre-built models or reference databases. So the results that you get from these kind of tests are only as good as the model. For example, if a honey has not yet been included in the database, it may yield a false positive result for fraud. And this is not because it's fraudulent, but just because the particular botanical and geographical origin of that honey has not been captured in the database. So if we had to use this method, it would require a very comprehensive database, and the feasibility and practicality of developing something like this on a global level is questionable. Molecular methods can be used to confirm botanical origin by analyzing pollen DNA in a honey sample. However, the success of these kinds of methods depend on pollen presence. They're generally not well suited for geographical origin determination. Elemental methods are also useful for determining geographical origin and may be used for botanical origin too, depending on pollen presence. And this is because minerals in a sample of honey mainly come from the nectar and pollen grains, and metals in the honey sample are derived from the soil. So you can trace that back to geographical or botanical origin. So which of these methods would be most suitable for honey origin testing? If we look at pollen analysis, it gives us good information on botanical origin, but generally requires some kind of supplementary method because it's seldom that you get a honey that's composed completely of one type of pollen. There's always traces of different pollens, so you want some kind of supplementary method to back up your results. Chromatographic methods also give us very useful information on botanical origin. They generally can stand alone and don't need some kind of supplementary method, but they have very limited use in terms of geographical origin. Isotopic methods are useful for both botanical origin and geographical origin. However, they generally aren't used alone. They require some kind of supplementary method to verify results. Spectroscopic methods give us useful information on both botanical and geographical origin, and this method can generally stand alone and doesn't need supplementary testing. Molecular methods are useful for botanical origin if pollen is present and doesn't need any kind of supplementary method. And elemental methods can be used for both botanical origin and geographical origin, but their uses for botanical origin are limited to the fact that pollen needs to be present. So if we look at all of these different techniques, we can see that spectroscopic techniques tend to be the most promising for determining honey origin. However, this depends on the completeness of a database. So to bring this all together, there is great variation in honey due to geographical and botanical origin differences. This variation would need to be captured in any kind of model or database that would be used for honey authentication. Spectroscopic techniques appear to be most promising for honey origin testing. However, this being said, the feasibility and practicality of collecting representative honey samples across all geographical locations and plant sources is very questionable. It would be really hard to basically collect every type of honey that exists in the world. Another potential solution would be to combine other techniques, but this means more testing and therefore higher costs. There is, however, definitely a gap here that needs fulfilling. This is very clear. So either with an existing technique or with a completely new approach. But scientific communities need to work together to come up with an answer in order to protect the livelihoods of our beekeepers and the value of our honey industries. Thank you.